I helped Steve Isles run the mass spec core facility, and we're here was accepted the invitation to come and share some studying we met for a while. He currently works at SCIEX as a development for life science research, biopharma application. Bachelor's in chemistry at Carleton College in Northfield, Minnesota. His PhD developing protein derivatives for enzyme replacement. Doctoral work at the National Institute using mass spectrometry-based functional genomics at the University of Minnesota, where he also worked at the genomics core facility, where he was able to different.
All right. Yeah, no, thanks for hosting me. This is a project that I've been involved with over the last year or so that I'd like to share with you all today. And I'm going to sit down so I can use my run around my screen here. But essentially, I'm going to share this data, a piece of data right here. And this is something that's commonly seen in the formulation of mRNA. In blue here, you have a free or naked mRNA here that's highly pure. But once this mRNA is formulated or encapsulated within a lipid nanoparticle, all of a sudden, these peaks over here to the right start to show up. And this is a reverse phased chromatogram here. So these peaks here to be at higher increased hydrophobicity. And, for a while, there's a mystery of what these peaks are. So, for example, is this aggregated mRNA? If you do gel electrophoresis or capillary electrophoresis, size-based techniques, these species did not show up, so there wasn't a size component to it or a significant size component to it. The lipid matrix effects, no residual lipids are detected. Carryover, not observed in blanks, only in the injected sample. So again, this is, what is happening? Why are these peaks showing up? And just to go backwards, obviously, we just had the COVID pandemic, and we're fortunate enough to have, a couple of the vaccines from Moderna and Pfizer, to immunity to COVID and different COVID variants. And, this is really, brought out the promise of mRNA as either a vaccine, obviously, or as a therapeutic. And there's several advantages or benefits of using mRNA as a therapeutic. You're using the ribosomes to create the active product, the protein of interest that you're trying to express, and so you get amplification through this process. The mRNA can be targeted potentially to a specific site. So there's a precision aspect or precision potential of using mRNA. You can create a sequence and then have it transcribed. So basically any sequence can be created very quickly. And all of the translation occurs in the cytoplasm with the ribosomes. So there's low risk of any DNA incorporation or genotoxic types of consequences. mRNA is transient. And, when you consider other biologics, like protein-based biologics, it's relatively inexpensive to manufacture. You're not using bioprocessors and host cells and all those types of mechanisms that have costs related to it. The biggest cost to manufacturing or producing mRNA, I believe, is the in vitro enzymes for transcription, mapping. To deliver genetic material or mRNA material, it is lipid nanoparticles are commonly used. So this is an artist's representation of what a lipid nanoparticle with mRNA encapsulated within it may look like. And typically, there are a few different lipids that are used in these formulations. The first is the ionizable or cationic lipid, cholesterol species, helper lipids, typically like a phosphatidylcholine, as well as pegylated lipid. And these lipids, together provide different physiochemical properties of the lipid nanoparticle that affect all sorts of different functions. These molecules, these encapsulated molecules are very difficult to analyze. So there's numerous analytical challenges, and there's several different quality attributes that, in the development and process analytics that need to be considered for making a, material that can be delivered. So, for example, specific lipids and the combinations or the ratios of the lipids that are used in these formulations, they can affect all sorts of different, properties, circulation lifetime, encapsulation efficiency, endosomal escape, the size of the lipid nanoparticle, toxicity, so on and so forth. The temperature, remember that the COVID vaccines had to be stored at these very low temperatures to be effective. So there's lots of different considerations when you are making these types of formulations, and there's no, this is still a nascent science here. There's no tried and true way of so I want to focus specifically on the ionizable lipid. So a typical ionizable lipid, this is this structure here is MC3. MC3 is a lipid that's used in the formulation of Onpattro, which is Onpattro is an siRNA drug, and so the MC3 is the ionizable lipid that's used in the LNP to deliver Onpattro, or the siRNA from Onpattro. A couple of features of a typical ionizable lipid would be, the head group here that has a tertiary amine. This is where, the positive charge would charge. Typically an ester group here, as well as, some kind of olefin or alkane type that would contribute to the particle. And there's the ionizable lipid has several different properties. So for example, they can, depending on the structure, it can impact target delivery, endocytosis, as well as endosomal escape into the cytoplasm. They also can have adjuvant effects, in the case of vaccines. So ionizable lipids are extremely important within LNP formulation, effectiveness, vaccines, or other therapeutics. And, down here on the bottom here are, different structures of ionizable lipids. There's this is MC3 again that has desaturated alkyl groups. There's multi-chain ionizable lipids, branched, polymeric, biodegradable. These types of lipids are designed on purpose to be biodegradable, so they can be removed, from after administration of the biological system. So here are the formulations, the lipids that are used in the formulations for the two different COVID vaccines from Pfizer and Moderna. And one thing I just wanted to point out here is that the ionizable lipids, that they constitute about half of the total lipid material amount within a lipid nanoparticle. So the Pfizer lipid uses ALC 0315. The Moderna lipid is SM102. both have that, that characteristic tertiary amine, as well as different branched aliphatic chains. So about, I guess this is December of 2021, so a little over a year ago, Moderna published this paper in Nature Communications, and they saw that they were observing a loss of mRNA activity or loss of translation of mRNA material within lipid nanoparticle delivery systems, and they proposed a mechanism for why this is occurring. And what they proposed was that the cationic or the cationic ionizable lipid was undergoing N-oxidation, and this N-oxidation was leading to degradation or decomposition of this lipid species down to a secondary amine as well as an aldehyde. The aldehyde is a very reactive species, and electrophilic type of species that could potentially react with the bases within. So essentially, they proposed that this mechanism was the cause of these that are eluting, later within this reverse phase chromatogram. So they proposed that lipidation was occurring on the mRNA, and the lipidation, if you covalently modify mRNA, the ribosome can no longer translate it. So you basically have a loss of function of the. So this is a very big quality concern, and these rates are highly dependent on structure. They're complicated to study, and if you look at an ionizable lipid preparation, the challenge is that not all ionizable lipids have a risk associated with them. So there's other peaks here that are impurity peaks that could have oxidation at other parts of the molecule. So some of these peaks may be more problematic than others. So now on to mass spectrometry. Mass spectrometry of lipids is fairly difficult with conventional techniques. Here is an MSMS spectra of a component here at, or a precursor mass of 788.61. This is in positive ion mode, and what you see here is you get this 184 peak, and that is diagnostic for a choline head group. We have the precursor mass, so we know that this is a phosphorylcholine species. From the precursor mass, we can deduce that this is a 34-1 some composition, but other than that, we really don't know a whole lot else about this, about the structure of this phosphorylcholine species. So about a year and a half ago, a newer technology came out that from SCIEX on the Xenotop 7600 instrument that was released at ASMS the year before last, so about a year and a half ago. And this instrument offers an alternative fragmentation that we call electron activated dissociation, or EAD for short. And this is a mirror plot of that same CID spectra I shared in the previous slide of that phosphorylcholine species, and with the EAD-based fragmented spectra on top. And as you can see, the EAD spectra is a lot more information-rich. So if we look a little closer at some of this data, we still have that 184 peak. So we're not losing any of the information from the CID spectra. We can still tell that this is a phosphorylcholine or a choline head group. If you look at this peak next to it, this doublet at 224 and 226, This tells us that the backbone is a glycerol backbone. If there was a 225 peak there, that would indicate a nitrogen atom, and that would be a sphingosine backbone. So we can infer here that we have a glycerophospholipid, not a sphingolipid. And then if we move along down the MS-MS spectra here, this peak here at 479 is a fragment between this ester group on the glycerol backbone of the SN1 position. So this 479 is diagnostic of an SN1 position, and from the molecular weight of that acyl chain, we acyl chain, so the SN2 would be 18-1. So we can get regioisomerization distinction from this technology. And then all of the other fragments at the high mass range of the fragment ion spectra, if we start here with the precursor, the difference between the precursor and the next fragment ion here is 15. So we're now fragmenting these carbon-carbon bonds from the acyl chain itself. So 15 and then 14, we're basically fragmenting all these methylene groups here. This is one of the big advantages of this technology, is the ability to efficiently fragment carbon-carbon bonds, where other fragmentation technologies, are not as efficient or can't really fragment carbon-carbon bonds at all. So from this information, we can walk through the mass spectra and determine, once we see a difference of 13, that's where we can figure out where a double bond is localized, or at least where the position of it is. And then based on the relative intensities of the fragment ions around that double bond, we can also infer whether it is an assist or single MS-MS experiment. We can completely characterize the structure of a glycerophospholipid. No other fragmentation technology can provide this amount of information within one experiment. So, how are we doing this? Again, this is using what we call electron-activated dissociation. This is a picture of the 7600 here. It is a quadrupole time-of-flight instrument. The inset here is a blow-up of the collision cell, or I should say collision cells, because there's two collision cells. There's a traditional CAD collision cell, and then in the front here is the EAD collision cell. These black end caps here are housings for electron filaments. The electron filaments emit free electrons, into the EAD collision cell for free electron-based fragmentation. A couple of unique features of this device versus other commercial electron fragmentation techniques. Number one is that the transmission of free electrons to the collision cell is near 100%. So we're shedding a very bright electron beam onto the precursor ions, and this enables very fast, efficient, and reproducible electron fragmentation. So, for examples, for peptides, we set reaction times at 10 milliseconds. For singly charged species, reaction times take a little longer, but no more than 30 milliseconds. The other unique part of this device is that the user can input different electron kinetic energy values. So traditionally, when I've used electron fragmentation, it was doing peptide or proteomics type of work, and ETD or ECD commercial devices are limited to really fragmenting multiply charged species like proteins and peptides. It's very useful for determining sites of post-transition modifications, for example. So certainly this device can do those types of experiments as well. But again, we can increase the kinetic energy, and right around 8 electron volts or so, the mechanism switches from electron capture dissociation to electron impact. Kind of what you would find in a GCMS, but again, this is within an LCMS instrument, and so this is where we use somewhere around 10 to 15 E fragmentation of metabolites. All right, so we were able to we acquired an ionized lipid here. This is just a raw material, and this is this MC3 ionized lipid from the Ampatro drug from Alnylam. And this is just a comparison of a CID-based fragment spectra and an EAD fragment spectra on the bottom. So just at first glance, you can see that the EAD fragment spectra is just providing a lot more fragment ions, a lot more information that could be used for structural elucidation of the species. If we look at the high mass range of that fragment ion spectra, this is kind of what I did, or I walked through with that glycerol phospholipid example. But within these alkenyl chains, we can start assigning different structures to these fragment ions. The differences here of 14, 14, and then all of a sudden 13, 13, 14, and then 13, 13, 10. And essentially, we can use this data to confirm this alkenyl chain structure with double bonds at position C. The other fragment I want to point out here is this 511.5. So if we see an impurity or a mass shift, we can use some of this information to help to guide us on where that modification resides within the structure. So, we were able to confirm the structure of the main peak MC3, but what we really wanted to get at was, can we determine the structures of some of these impurities that we also see within the raw material? So I've listed these as impurity 1, impurity 2, and impurity 3. There's several other impurities within this raw material formulation or substance. But if we focus on impurity 1 and impurity 2, these are earlier eluders in a reverse phase chromatogram. And we can see from just the top MS scan, these have the exact same molecular weight. And this molecular weight is exactly 15.995 mass units above the main peak MC3. So we know that the oxygen, the mass defect of oxygen is 15.995, and so this suggests that we have oxidation or molecular oxygen incorporation within this MC3, causing all these impurity structures. The question is can we pinpoint the location of oxygen incorporation within these two impurities? So if we start with impurity 1 here on top, this 132 fragment ion was common to the main peak MC3. And so that indicated that oxygen incorporation was not within the head group here. But when we looked at some of the fragment ions at the higher mass range, we did see some differences. And right here, so these fragment ions are listed here at 558, 557, and 587. These were unique. We did see disruption of the double bond at position 6, and then oxygen incorporation after that with the 574 and the 587 peaks. And so this indicated that we had that oxygen incorporation was somewhere at position 6. We drew an epoxide structure because that seemed to fit the data the best. Conversely, with impurity 2 down here, instead of this 132 peak, we now saw this 148 peak, so plus 16, and that indicated that oxygen incorporation was somewhere within the head group. And then we also observed the 61 peak here, and the 61 peak could only be explained if oxygen was being incorporated within the tertiary mean. So from this information, we drew the N-oxide structure, for this oxidative impurity. So going back here, we were able to distinguish and characterize these two structures. Impurity 2 was the N-oxide species, and so this one, could be the more problematic impurity that if you're formulating these types of mRNA therapeutics that you want to keep track of. I also want to talk about impurity 3 over here. So this is a later eluting peak, and we overlay the main peak MC3 data in red with this, so this impurity 3 showed a mass difference of 2. which indicated, addition of 2 hydrogen atoms. So it appeared to be a saturation impurity. If you overlay the 2 fragment ion spectra, you can see this +2 mass shift at the high mass range. So if we fragment, for example, we fragmented one of the carbon-carbon bonds over here, this would, no matter which chain was fragmented, it would contain that saturation at C6. However, as we continue down the mass range or down the chain here, you eventually get to a point where you have instead of just 1 species, you have 2 species within the saturation impurity, and so you have kind of equal probability of fragmentation over here, or either containing the saturated peak or the unsaturated. The saturation. The saturation point or you get these 2 doublets of the blue species, indicating a mixture there, and from this data we can infer that that saturation indeed at C6 or at C. So, taken in sum, this is looking at the some of the different impurities that we found within this raw material, and comparing this to, we calculated relative abundance compared to the main peak MC3, and this is all done from MS1 data from the XIC peak, and this is relative abundance. Certainly some of these efficiencies, but these are the ones that we characterized using the EAD fragmentation. We also observed lower than this, but just for the sake of time, we didn't. But one other thing I wanted to point out that's unique to the instrument is with a conventional quadrupole time of flight instrument, ions are generated at the source. Their ions are focused and they traverse through the various devices within the system, and they reach the time of flight region. Once they reach the time of flight region, a high voltage push is applied, and a slice of that ion beam is taken for mass measurement. During mass measurement, ions are still being generated and still going through the. They're not, they're being lost. They're not being sampled, and this is the trade-off that you typically see with a high-resolution instrument, where you get high resolution data but you lose sensitivity. So with this instrument, we have an ion trap that sits behind the CID collision cell, and this ion trap collects ions and then releases them in a manner that's synchronous to the time of flight pusher, and thereby avoiding the loss of these ions and maximizing MS sensitivity. So the electron fragmentation that we're generating, hundreds of peaks with electron fragmentation, but to take advantage of all those peaks, because we're really diluting the ion current down after fragmentation, to take advantage of the peaks that are generated by this type of fragmentation mechanism, it is important to really maximize MS sensitivity. So, I'm going to share, I want to share one other example of an ionizable lipid. So we also characterized ALC0315. Again, this is the lipid that's from Acutis that was used in the Pfizer-BioNTech vaccine. And this is comparing, or this is a CID mass spectra of the ALC0315 component. So again, CID, it just doesn't fragment lipids very well. From this data, we only see about 7 meaningful product ions. So we do get some structural information, but it's hard to draw a complete picture, with CID information alone. If we look at the EAD fragment ion spectra, again, we're generating a lot more fragment ions. Instead of those 7 or handful of fragment ions that are generated, we're now generating over 100 that can be used to help elucidate structure. And, just to point out a couple of them here, so there's this 427 fragment ion that fragments as nitrogen carbon bond right here. There's this 172 fragment ion right here. That kind of, between the 427 and the 172, we can use these fragment ions to, kind of segregate that head group, and we can look for oxidative impurities there specifically, again, segregate them from other parts of the molecule here. And then there's this 528 here, fragments the ester bond. So this is the pure ALC0315. If we start looking at impurities, so here is an impurity right here that showed oxygen incorporation, and the fragment ions that we see here at instead of the 171 and the 427, we now see a 443 and the instead of 171 and 427, we see 187 and 443. And so from this information, this suggests that oxidation is occurring, for this observed. And then furthermore, we also saw another oxidative impurity. Instead of that 443, we saw 441. So it was oxidation minus 2 proton or 2 hydrogen atoms. And then instead of the 185, we see or 187, we see 186. And then we also saw this fragment ion here at 735. And the 735 could only be explained if that oxidation and loss of 2 hydrogen atoms was a carboxyl. Instead of an oxidation that we described previously, this here was a oxidation to a carboxyl. So again, we can use this information to help deduce, whether it's problematic or less impurities observed. All right. So from here, I'm going to kind of switch gears and talk about the targeted methodology. So that's what I shared was more characterization, more impurity identification. This is characterization using targeted methodology on what we call the 7500 system, which is a triple quad type of system. So, I mentioned this before, but it's one thing to characterize some of these impurities, but then once you have that characterization information, now you can start to quantify some of these types of impurities. And this would be important for monitoring breakdown products, degradation products from these, either degradate or lipidation of the mRNA or breakdown of the actual lipid. So SCIEX has had a long history or tradition in lipid type of analysis, and we've developed methodology to profile up to 20 lipid classes that contain over 2,000 transitions. So the lipidome is a very complex, or lipidomics is a very complex field. There's several co-eluting lipids within a biological sample, and so being able to have technology to monitor as many lipids as possible help. So this methodology, again, we're targeting 20 different lipid classes, either positive ion lipids or negative ion lipids, 900 or 1280 grams that are associated. So this methodology can then be taken and applied to lipid nanoparticle or liposome. Here you see a chromatogram of the helix separation. So we are separating based on the head group to help reduce the complexity of a lipid mixture. And then within each of these chromatographic peaks, we can schedule MRMs specific to different, different chain lengths. In the case of liposomes or lipid nanoparticles, one of the breakdown products it would be like a hydrolysis to a lysophospholipid. So using this base methodology, we can then look specifically for lysophosphatidylcholine species. We get really nice reproducibility, with this type of instrument, and we can take this method with 2,000 or so MRMs and then filter it down to just what we want to look for, depending on the formulation. And then certainly you can build in the proprietary or steroids that you also would, other undesirable species. So kind of a nice starting point to build these targeted quantitation methods. This work was done. This is a picture of the 7500 triple quad instrument. SCIEX offers other types of triple quad instruments as well. This happens to be our high-end platform. But just want to emphasize methodology to do this type. And just to summarize what I've shown here today, the impurities that are part of lipid nanoparticles, they can affect the integrity as well as mRNA activity. They've been implicated in, or the oxidized species of some of these ionized lipids have been implicated in covalent modification of mRNA, rendering it non-translatable or inactive. And when you think about an mRNA species, it's something that would be 2,000 or 4,000 bases. It doesn't take a whole lot of oxidized material. I mean, you only need one reaction to occur, to render that material inactive. So as a result, it's important to be able to monitor very low abundant. So identification of the impurity species is important, obviously, so we know which ones may be more problematic versus less problematic. But certainly with trying to measure things at low abundance and characterize them, with a high degree of certainty, is difficult with existing technologies, and so this is the applications that this EAD help with. Finally, with quantitation, to ensure raw material and LNP quality, understanding the relative percentages, components, and purity profiles. So, this is just kind of the beginning of the story here. This field is really, from that Moderna paper, there's been a lot of interest, for the characterization of these covalent modifications. So, we've demonstrated that we can characterize the structure of lipids in great detail, but, what we'd really like to know or have a better idea of is what is the exact mechanism of that covalent modification. So Moderna proposed, the N-oxidation being degraded to an aldehyde species, but, that's kind of a hypothesis. It'd be nice to be able to use this technology to isolate a modified nucleobase, and to use this technology to get at the exact structure of the lipid and where it is covalently modifying the nucleobase. If you have this type of information, this infers mechanism, and then you can take steps to mitigate, at least in drug development or drug formulation strategies to avoid lipidation. And some other questions are there specific nucleosides that are more prone to modification? You hear different perspectives from mRNA-based therapeutics. And then does the mRNA structure itself, does that influence covalent reaction? We know that that RNA has to be a certain length. It has, like shorter RNA species that you'd use for CRISPR, for example, like 100 base pairs, that does not seem to get modified. So there's a dependence on length, and there probably is a dependence on secondary structure, or proximity. You know, perhaps if nucleosides are in a certain structure, their pKa's or, you know, their nucleophilicity changes, and that could influence COVID. These are definitely areas that I'm curious about, and, you know, hopefully we can access the samples. You know, and a couple of closing remarks here. SCIEX is part of Danaher, specifically the Danaher Life Sciences organization. And I did want to, you know, so here's different companies that are part of Danaher Life Sciences. I did want to mention Precision Nanosystems in particular. So Precision Nanosystems, they're part of this Paul Cytiva group here, and they make devices to formulate lipid nanoparticles. So, you know, it's great having, you know, other operating sister companies that we can work with, you know, to provide materials and, you know, to give us ideas for some of these experiments. In particular, I want to thank Adam Crow and Nikita Jain. We got the ALC 0315 samples in collaboration with Acutis, so I want to thank Evan LaRay. And then here are all the folks at SCIEX and the samples, or, you know, yeah, I mean, with that, thank you for your attention. Hopefully this was interesting, and I'd be happy to take any questions.
Thanks so much. Help yourself to more of the treats before you start off. I did EADs to the